BACE is a group that is meant to bring together diverse peoples with broad interest in epigenomics to just learn, share their knowledge, and if possible, find opportunities related to epigenomics. There is, as you probably know, a meetup page, and there's also a YouTube channel that has all the talks from past events. So this is why we're recording this, and it will be shared after the event. Yeah, today we have two speakers. Oh, I guess before I say that, as I was mentioning that earlier, I am not Monica. The organizer of the event is Monica Malesewska, who is the kind of leader of the BACE group. But today she had a last-minute work-related conflict, so she asked me to host this. So my name is Sophia. I currently work at Arsenal Bio, and I will be hosting this event tonight. So tonight we'll be hearing from Yihui and Karin Nair, starting with Yihui, who has a PhD from UC Davis and is currently working as a bioinformatics scientist at 10x Genomics, and she has a background in studying the epigenome and transcriptome of autism. So with that, I will stop sharing. And Yihui, feel free to go ahead.
Thanks for the great introduction. I will start with share screen. Thanks, Sophia, for the great introduction, and thanks, Monica, for bringing us to this event. And I'm happy to share with my PhD research on the placenta research, especially the placenta with autism research on epigenetic signatures. So a little bit about me, for those of you that not know me quite well, I just graduated from my PhD at UC Davis in genetic and genomics. And at UC Davis, I did my research in Dr. Janine Cell's lab, working on prenatal epigenetic signature for autism spectrum disorder. Currently, I'm at 10x Genomics as a scientist applied bioinformatics, working on the single cell relative research. Feel free to jump in if you have any questions relative to my research. Right, and the reason I want, I talk with Monica about this opportunity to share with this research is my paper with my PhD research just out. And please feel free to check out as Genome Biology for our latest publication. And thanks for all our collaborators for this research. So I will tell you more about my research on placenta epigenetic signature with autism. So autism is short for ASD, short for autism spectrum disorder. It's a neurodevelopment disorder. Children with autism will show repetitive behavior, language deficiency, and communication impairments. Right now, there's 1 in 54 children in the United States affected by autism. It is a pretty complicated disease because there's no currently molecular diagnosis test and there's no drug cure. Children will, the only test can be diagnosis with autism is clinic diagnosis at 3 years old or above. And the early intervention can really help children with better development gains. So we're trying to see whether we can find early stage markers or early stage signatures that we can predict children's behaviors in later stage. That can help a lot for the family and also children. As we know, autism has both the genetic and environmental factors. Famous twin study shows that there's a heavy genetic component with autism. If one sibling is having a diagnosis as autism, the other sibling will have a much higher chance to diagnosis as autism as well. Other environmental research shows that prenatal vitamin intake during the first months of pregnancy is pretty critical for children's neurodevelopment, especially for those large longitudinal studies. They replicate and show that autism is really relative to nutrients and environment factors. Let's look at this pie chart. The majority of them still are known, although there's a lot great proportion with a single gene mutation, common structure, and de novo gene mutation, but there's still a lot of unknown. So my lab at UC Davis focused on the epigenetic research to show the field for both the genetic and the environment factors. For me, for my personal research in UC Davis, I specifically focus on DNA methylations. Although there's a lot of epigenetic signature that in the field, my research is focused on DNA methylation. And I'm using a very interesting tissue that is less studied in the field. It's human placenta tissue. Why we use placenta? Because it's an easy, accessible, and non-invasive tissue compared with other tissues that we can not easily access during pregnancy or right after pregnancy. Placenta is an easy tissue compared with brain. And also it shows the same in utero environment as fetus. It can be used as transfer nutrients, exchange gas and waste, and also protect fetus from infection. The most important is that placenta show the similar methylation pattern as oocyte and the pre-implantation stage. The only non-cultured tissue containing partially methylation domain is placenta tissue. So my research is especially focused on placenta for this topic. We have two prospective longitudinal studies involved in this research. The first one is called MARBLE. MARBLE is short for Markers of Autism Risk in Baby Learning Early Sign. We have one collection site at UC Davis. The other study we have is called EARLY. EARLY is short for Early Autism Risk Longitudinal Investigation, which including 4 collection sites, but each site with less samples, and across different parts of US. Both studies are prospective study on enriched cohort, means we have the children and family involved in this study already have an affected sibling. So the next sibling will have a much higher chance of getting autism. We separate the children into 3 groups, into autism, typical development, which is our control, and if not fit with the criteria for either autism or typical development, which will character as non-typical development. We recruit mom during or before pregnancy, and after during pregnancy time, we collect their questionnaire data like what they eat every day, and we also collect biological samples like blood. And the parents also fill in all the forms and all the other information. After pregnancy, we collect placenta tissue, store them in -80 until their children reach 3 years old. We bring them back to the Mind Institute for the final diagnosis. At that time, the UC Davis Mind Institute will have physicians to work with the children for their development assess. They will be assigned to 3 groups for those samples. Previous research I did, we found there are 2 interesting markers, CYP2E1 and IRS2. One is affected, one methylation pattern is changed by the diagnosis group, and is not affected, is affected by genotype. The other one is affected by whether take prenatal vitamin or not during the first month of pregnancy. So we already found some interesting signatures using placenta tissue firstly. So we decide to move on to a more complicated study with more sample size. This is a brief outline for our sample this study. We have discovery group separate into the into marbles and replicate into earliest and additional marble samples. In the discovery group, we do differential methylation region analyze, identify the 22q13 block, and we replicate using additional early samples and marble new samples as internal replication. These are the methylation part. Besides that, we also look at the genetic signature on calling variants on the same sample using whole genome sequencing. For the epigenetic signature, we're using both the whole genome bisulfite sequencing and the epic array. And in the whole genome sequencing, we identify the SNPs, CSVs, and we also validate our result using autism postmortem brains. After we do this population study, we validate our result using cell line after we identify this NHEP gene, which I will going to cover in more detail in later slides. We overexpress that in cells, and we also see the response with oxidative stress. In total, we have 204 subjects from 2 prospective autism cohort. This is the result from this discovery group. In the discovery group, we compare autism vs typical development, which is our control. We adjust for sex, cell type, and performed 100 permutation tests. 134 DMRs passed permutation significant, which mapped to 183 genes. One of them passed the genome-wide significant. You can see the red dot here. This is the only one that passed the genome-wide significant with our stringent cutoff. But I also can notice that there's 12 DMR on the same block also passed the raw permutation, raw p-value significant. And the other 12 DMR is located on the same block, 22q13 region. It forms a 118 base pair 118 kb hypomethylation block. So those are our interesting findings to begin with. After that, we also look at what are those differential methylation region is doing. We find out this is a gene ontology analysis. We find out it's enriched at histone acetylation for different histone markers, chromatin organizations, and we also find that it's actually enriched for fetal brain enhancers. This placenta markers is also enriched. So because we use placenta tissue to do the discovery group, so it's kind of like we can see our findings in placenta, but it's very interesting. We are hitting fetal brain enhancer as well. So our placenta markers do hit the brain markers as well. After that, we look at, because we saw those 12 interesting differential methylation, we're wondering we found out they are highly correlate with each other. See this plot, we see the Pearson correlation score for the correlation between those 12 DMRs are pretty high. Besides, we identify the differential methylation region in the discovery group using 19-2 samples. We also validate our result in the other replication group early, and especially we also validate our result using a third population in the marble group. And you can see autism has lower percent methylation than typical development. And the gray group is in following between. So autism has lower percent methylation than the lump typical development, and the lump typical development has lower methylation than the typical development. So this following lies with our expectation, and we are very excited about this 22q13 result. Because yes, we are sequencing different sequencing platforms, so we are not combining them, we're using them as different replication group. After we find this interesting 22q13 region, those are the 12 DMRs that highlighting the black box. We're wondering what exactly is inside that region, because we couldn't find any protein coding gene inside this block. After my previous PI, Dr. Janine Nassel mentioned, I load those tracks from the ENCODE data. We found out there's actually an active H3K4 trimethyl peak, showing that there's an active promoter here. After I look in deep, and it's showing up in the different oocyte, placenta, and brain samples, and they showed variable between samples. After I look deeper, found out there's a new gene, there's a non-coding gene, and we rename it as NHEP. And this gene is our first time to identify it, and it's being approved in the gene database. After we find this NHEP gene, we want to look at what tissue it expressed in. As you see, this is from GTEx database. We find it expressed differently in different brain regions. And it's pretty variable. For example, cortex has lower expression, and for some other cerebellum and other tissue, hippocampus have different expression as well. So it does variable between different samples. We also want to characterize more about this NHEP gene, and we find out using different tissue and find out NHEP gene have different mRNA level among different tissue. And this NHEP gene is conserved in primate only on the DNA level, and it's pretty highly expressed in both fetal and adult brain. And we also show a significant difference between autism and typical on the placenta sample.
Sorry, Yihui? I don't know if you might have covered your microphone. You sound a little muted.
Oh,
there you go.
Is that better now?
Yeah.
Oh, cool. Sorry. Yeah, so this slide is showing that the mRNA level for NHEP is in the same trend as the methylation level. And we also show that NHEP level expressed in different cell line. And it's show we find out the LUMI, which is a neuronal cell line, has been shown as difference in differential LUMIs and undifferentiated LUMIs. So, and HEC 293T has the lowest expression of the NHEP gene. After that, we also want, because we want to understand the NHEP gene more, and the placenta is a very sensitive tissue during pregnancy. We want to ask whether the environmental exposure, especially the oxidative stress, whether related to that. We find out it's only oxidative stress is only affect NHEP level if it's differentiate LUMIs. For the non-differentiated LUMIs, it doesn't change the cell variability, but in the differentiated LUMIs, it changes the cell variability. And the different, we can see the differentiated LUMIs actually show different response to oxidative stress levels. And we do a longitudinal test on the differentiated LUMIs in the NHEP level change with whether it's a differentiated LUMIs levels or not under oxidative stress. And we're also able to recover that after removing the oxidative stress. Besides that, we also find the cell overexpress the NHEP shorten the doubling time. So the cell with overexpressed NHEP in the cell line, we see the doubling time is much shorter. So cell divide much faster. For example, the normal wild type cell divide for the 293K cell divide every 24 hours, but after overexpressed NHEP, it divide every 20 hours. You can see later down the stage, the cells with NHEP overexpressed divide much faster than the wild type. After because we find this NHEP gene, we're wondering whether it shows on the database is a long coding gene. We're wondering whether there's any peptide relative to that gene. And my labs were Dr. Jin cell labs with a great postdoc that work with me, Antonio, has found out there's OZAC sequence inside. So we clone that into a cell. And after that, you can see that this is this is showing the NHEP peptide with overexpressed. This is a fluorescent showing whether the transfection success. And this is a combined fluorescent to show that we have successfully NHEP expressed in the cell. So NHEPs do function as a peptide function that contains the OZAC sequence, which is conserved sequence. And we also find that it's in the nuclei rather than the cytoplasm using other fluorescent markers. This is done by Beck Yasu in the cell lab. And this is the NHEP peptide sequence I'm showing here. Besides all the functional analysis, we are also wondering if there's any genetic signature that can change methylation that we're showing the 22q13 hypomethylation block. This is NHEP that located here. And those are the 12 DMR that I mentioned identified by the differential methylation region analyzed. we find out there's an upstream insertion that's significant associated with the methylation in the 22q131 block. And we also that because we're using short read sequencing, Illumina read sequencing to do whole genome sequencing, so we find this insertion. We also validate our result using PacBio long read sequencing. We also can see this 1.6 kb insertion as well. This is what showing that the methylation here is significant associated with whether have the insertion or not. For example, let's break down this table into these are the samples in the typical development. We don't see a significant difference between whether carry this insertion or not. But in the autism sample, we see a significant lower percent methylation in the samples that have the insertion compared with not having it. So this is almost 1/2 of the samples, rather than this not have the insertion is almost the same. So we found out this insertion also changes the methylation as well. Besides that, we also look at whether there's other environmental factor, like who I mentioned previously, prenatal vitamin during the first months of pregnancy. We found out if mom take prenatal vitamin during the first months of pregnancy, will have a higher percent methylation than not take it. And when we break down by different genotype, only affected when when children carry the insertion. It show a protective effect on those ones that carry the moms that take prenatal vitamin during the first months of pregnancy. And we have, because we find this is kind of complicated, including a lot of factors. So we also want to, whether we can figure out some models or explaining why it has happened. This is our NHEP gene located here. This is the CpG island, and you can see there's a multiple tissues that show the yellow and red means the active promoter. Multiple tissue have the active promoter here. And actually there's 2 CTCF sites surrounding the insertion regions. And both Lumi cells have this CTCF site. This site with the distal CTCF is variable, so not all samples have it. And this site also is variable, and only brain sample have it and placenta sample have it. So we see that it's very interesting that the CTCF maybe plays an important role here. And we have a hypothesis model that is in progress of testing. There's 2 CTCF binding sites, and this is a fetal brain enhancer. This is our NHEP level. When the NHEP level is high and with higher expression model, those are the normal sample protected from autism, with the presence of 1 or 2 reference allele, and with moms that take prenatal vitamin during the first months of pregnancy. But if there is under oxidative stress and not take prenatal vitamin during the first months, it will bring this loop will bring. It will lower the NHEP expression and with lower percent methylation as well, with this long insertion pieces. So this model is about this insertion maybe disrupt the fetal brain enhancer promoter interaction, thereby reduce the response of the NHEP expression to the neuronal differentiate and the excessive oxidative stress. And the early prenatal vitamin maybe is can be used as a counter effect of the oxidative stress by adding methylation group and increase DNA methylation level at NHEP locus. This is still under testing now, but this is a hypothesis model we have from this study. Besides what we, any question?
Thank you, Lucas.
I see no.
So whoever joined recently, can you mute yourself?
Yeah, so those are the models that we took from the cell line. Because now autism is a neuronal development disorder, we also want to check and replicate our findings in brain. So we are fortunately also get some postmortem brain samples from brain bank. which we also validate our finding to see, and with the sample with insertion, we see a significant lower expression levels on NHEP than not have the insertion. And after we do also perform a differential expression analysis, found out there's almost 851 genes that significant associated with NHEP levels in brain past the FDR significant. And besides what we do with differential gene expression, we also do the genome ontology analysis. Find there's a lot of gene relative to oxidative stress, which further validate our finding in cell line using brain data and relative to dendritic development. Those are also, we also see cell functions in cell fate commitment and embryo development that validate in brain samples that are relative to NHEP levels. Because we also see a folic acid relative change, we also see a fatty acid metabolic process also related to that as well. Apart from those, what we done with the cells, with our expression cells, and we also the brain one, we also did a differential gene expression analysis with our expressed NHEP level in in 293T cell lines, which show a large scale transcription factor change. We saw almost 5K gene changed after we overexpress NHEP level in 293T. Before we talk about all the Lumi cell change with oxidative stress, we also see similar phenotypes that change in the 293T cell line. And those besides those we found in the Lumi cells, we also see cell cycle change and also brain disorder as well. And we also see some drug metabolism pathway change as well. And those are, besides, we find this interesting gene in autism, but we also see that it's highly enriched for Alzheimer and Huntington disease as well. So we're hitting some neuronal development gene. This is a brief, this is our overall recap of all the categories we compare with. We show the NHEP associated transcript in brain. This is a Safari, which is an autism database of autism risk gene. And we see the cell line changed NHEP gene with a comparison between overexpressed vs control. There's 30 genes in common with all the comparison. Those 30 genes show that this is a breakdown of the sublist of the 30 genes. We show those genes have chromatin functions and also have functions in histone modification and also racemic pathway and the dendritic function, and also can regulate some biosynthesis process. This is a take-home gene list for those people that want interested in learning more about autism and the NHEP gene. And this is an overall summary for the project I'm just covered. We're using differential methylation region to analyze and identify 138 differential methylation region, which different between autisms and using placenta samples. And we find this high confidential 118 kb hypomethylation block on 22q13 that replicate across different studies. And we find this interesting NHEP gene using H3K4 trimethyl from ENCODE, and we say that we show that it's highly expressed in brain, and also show that neuronal differentiate or oxidative stress can increase that NHEP levels. We find out the transient NHEP overexpress can increase the cellular proliferation time, can also change those synthesis and the neurogenerase pathways. And we find significant overlap with other known autism risk gene markers. Besides that, we also find a common structure variations that included in the data analyzed, and we also validate our research in brain sample as well. And with this together, this study takes the innovation approach to utilize placenta's tissue from a high perspective longitudinal cohort and find a novel autism risk gene that it responds to oxidative stress and with a common structure variation. So we found that there's some interesting about this project, and Dr. Jin in the cell lab is going to work on more on this gene and this research. So I'm happy to share this story with you guys. This is a acknowledgement slides. I have a lot of people want to thank for, and especially Dr. Jin Lascelle and my wonderful collaborator, and also funding resource as well, and also Antonio for doing all the cell line work. And thanks for your attention. Any questions?